So before we get started, I want to address some questions that were in the chat and see if we have anything from last time that you would like any more clarification about. Oh, there we go. All right, I'm going to address the very first question, which is if you clone the repo, how do you update it again? Which is an awesome question. Okay. If you have Windows, you would go to your command prompts. Okay, so if you're Mac, you would go to terminal. Make sure that you're seeing something that looks like this. Okay, so let's say that I have the previous version of this of the workshop repository. So first thing you're going to do, let's go ahead, go to our website, which is just bigbioinformatics.org. I'm going to put this in the chat. This is where we're starting to, we just really just put this together. And I'm putting in the chat so you can know where to get it. Basically, this is where a lot of our stuff is going to be from now on. And you can find it other places. You can find us on GitHub and YouTube, but this is the main place where I think people will want to access the resources that we're creating, because you have kind of a nice user-friendly way to see everything, right? So, so for module 2, the GitHub repository and slides are at these links here and here. So when you open the slide link, it'll actually download it to your computer. But okay, so let's say we're now at GitHub and we're in the GitHub repository for this workshop. Okay, so when you first go to clone this, what you can do is you can just copy this by hitting this button and then come over here and you can type git clone, and then you can get the, basically it'll just download the whole thing onto your computer. And when you look at it, so, okay, so where did it download it? Well, I'm in C users MILLE. Okay, so on Windows, all you got to do is go to this PC, go to C users MILLE. I don't know what it's going to be on your computer. It's definitely going to be different, but it's whatever your username is on that computer is where the command prompt opens up and where you'll be downloading it to if you don't change your working directory. And as you can see, it downloaded it right here. And I go inside and I see I've got everything here. Okay, so let's say that there's a change which occurs, right? Let's say that I don't have, for one reason or another, I've got an older version of this, and so I don't have module 2. So it's hard for me to actually simulate this appropriately because I do have module 2, and I'm not sure what the command that you would specifically use to roll it back to an earlier commit would be, but let's just say, for the sake of argument, that I will go ahead and I'll change something on here. Okay, so that's one way to do it. So let's say I change something. Let's say that I change this README and I added a line at the end that says, you know, print hello world. This line doesn't really serve any purpose. You know, this could be an example of some R code. Okay, so I'm going to make this change and I'm going to commit it directly to the main branch in GitHub, which means that basically I made the change and you can see now it's changed. And without it ever entering my computer, that change got made, which means that now there is a difference between the remote repository that lives on GitHub, right? Because it's got this new thing in it, and my computer, where if you go to the, this is a different folder, sorry. If you go to, right, if you go to what I cloned and we open up this readme.md file with, I don't know, Notepad, for example, you can see that it does not have that new information at the bottom. So how do I deal with that? How do I get the new version of the remote repository onto my computer? Well, the way is with git pull. So I'll go into that repository, single cell RNA-Seq workshop. And if you're not used to the command line, it's good to get used to it. So one of the things that I just did, which you might not have caught, is I started typing the name and then I hit tab to autocomplete it. Right, so I'm changing my directory to be the single cell RNA-Seq workshop, and I did that efficiently using autocomplete. So there I am now. Okay, So if I look at what's in this directory by typing ls, oh wait, apparently I don't have bash set up, so my bad. I just got a new computer, so I guess I haven't set bash up yet, but that's fine. It doesn't really matter because all we're really going to do is run one command, which is git pull. So git pull is essentially going to go out to the remote repository. It's going to find the latest commit. So this was the latest commit here. And it's going to pull the changes from that latest commit onto my computer. And you can see here that it's now finished. It basically said, here's what changed, right? It said you added 3 new lines to the file readme.md. One file changed, 3 insertions. And that's basically it. It said you added 3 new lines to that file. And now you're up to date. All right, so if you don't have the latest version of the repository on your computer, all you have to do to sync up with the remote is to go into that repository on the command line and run git pull, and it will pull that. Yeah, okay, good. Wes says you revert the hash to go back to last commit. I actually have no idea how to do that. It would be interesting if we could it's kind of out of the scope of this workshop to talk about Git, but it would be interesting to have like a Git cheat sheet that included some of those basic commands. Okay. And if you didn't want to do that, or if it wasn't working for one reason or another, you can always come over here and download this as a zip drive. And then you'll have the whole thing, you know, all you got to do is unzip it. And there you go. You got the whole thing right here. All right, so for module 2, we're going to be doing everything in this folder. So what I want you to do is go ahead and open up that R project file, just like I showed you last time. And this is where we're going to be doing most of the work today. You can even open up the module 2 R file if you want, and go ahead, if you haven't got these packages on your computer, go ahead and run lines 4 and 5 and make sure you get the packages. Essentially, this is a command that's going to automatically search your computer to see if you have the packages. If you don't have them, it's going to download them. Okay, so but while you guys run that, I'm going to address some of the other questions we got in the chat. Okay, so Mar sent a few questions. So question 1, when we look at the PBMC data or the 10x genomics data, we get an error message. And yeah, so I'll address that today. So question 2 is, I tried to load a count matrix coming from Geo, downloading a matrix.txt.gz, but then read 10x is not working. Yeah, so today, one of the first things we're going to do, actually, for the sake of time, I'm going to save this for the end so that people who don't want to stick around to watch this part can leave. But I'm going to show you how to get 10x genomics data and other kinds of droplet-based single-cell RNA-seq from a variety of sources. So including both the easy way from the 10x genomics datasets portal or the hard way from Geo, but probably the most realistic way you're going to get it. All right, so question 3 is, will you also explain how to process raw SRA files that we can download from Geo to get the count matrices and doing the alignment? I'm actually just about to describe exactly how you do that. So yeah, these are all great questions, and hopefully I will get to each of them today. Okay, so Mar made one really good point, which is if you use git pull, if you're using branches, which in this case we're not going to be doing, but if you are using branches and you have a modification inside one of the folders of your branch, you might lose your modifications. Okay, so that's a really good point I just wasn't aware of. I think it should, when you pull it, I think it should automatically handle that and preserve your branch, but I guess I don't know. I have never run into that issue personally. But it's a good thing to keep in mind. So don't use git pull willy-nilly. Like, you should really make sure you're understanding what it's going to do, and if there's an issue that you have with your branches, make sure that you look online and see if you can find a better explanation for how it's working so that you don't accidentally delete from your data. Okay, so Wes says if you pull from main, oh, okay, I see what you're saying. Yeah, so all right, so this is not supposed to be a Git tutorial, but in Git you have a concept of branches. So right now, if I type git status, I can see I'm on the main branch and my Git branch is up to date with the origin main. So that essentially this is telling me that there's nothing has changed since the last time I did a push, right, or a commit. So I can actually change to a new branch, or I can create a new branch. So if I do git branch, my new branch, and then I do git checkout, my new branch, it'll tell me this message, I switched to branch, my new branch. Okay, so what does that really mean? Well, let's make a change here. I'll just make, I don't know, let's make a new file. The nice thing about working in Windows is that when you change branches on the command line, your file system automatically tracks that. Or at least it always has as far as I've ever seen. So let's say I make another new change here. I'll add another new line to the README. So I'm just going to do this real quick. So I made this change inside of a branch, or at least I should have. So let's go ahead and do git status now. So I've made a change. Okay, well, apparently I made that change in the main branch. So apparently Notepad does not track that. My bad. Anyways, this is not supposed to be a Git tutorial, so I'm not gonna go any further on this. I guess it would be more of if you added a new file. So if I add this new file here like this, let's try that now. Git status. Okay, so apparently I don't know how this works on Windows then. Oh, right, because I'm in my downloads folder and not in my other folder. So let's go back to the folder that I'm actually supposed to be in right now, which is this one here. Okay. So in the command line, the mistake I made is that in the command line, I was in this folder, but in my file browser, I was in a different copy in my downloads folder. So I wasn't tracking the changes I was making. All right, so let's go ahead and do git status. I'm on my new branch right now. So let's go ahead and make a change. I'm just going to copy this. I'm going to make a copy and then type git status again. And now we can see that it's telling me I now have an untracked file. Let's zoom in here so you can see this a bit easier. I now have an untracked file, and it says readme/copy.md. So basically what happened was I made a new file, but I didn't register it yet with Git. So let's go ahead and do that. Git add. And I just typically write git add all. So that's what this means, git add all. So it's going to add all the changes that you've made in the branch that you're currently in. So it's going to register those. So let's go ahead and type git status again. And now we can see we have basically registered this. I think the proper term is staged. You've staged this. Again, I'm not a Git expert, but I know enough to be functionally capable with it. Now that you have registered this change, you can commit the change to the branch, to the history of the branch. So git commit, and you can write a message, made a copy of readme.md. So now I have made this new commit. Okay, so what is going to happen if I now push this back to GitHub? So I think that there is, okay, good. Yeah, so good. I was about to say, like, I don't think that's going to work. All right. go ahead and copy and paste the message it just said. Basically, it's complaining that in your, okay, I guess my understanding of this is that in the remote repository, this branch doesn't exist. And so if you're trying to push from this branch to the remote repository, you need to set this upstream branch as origin. Again, this isn't a Git like tutorial, but I think it's worthwhile to know like how do you actually do this on the command line. And so if something fails and then it gives you like this message, this typically will work if you just copy and paste that message. All right. So what's happened here? Well, essentially it's given me a message. It said, you know, we uploaded all of the stuff from the new branch to GitHub. And now it said, create a pull request for my new branch on GitHub by visiting this URL. So what does this all mean? Well, simply put, if we come over here, we can see that we now have two branches in our branch selector. We have main and my new branch. All right, so if I come to my new branch, I can see that we now have this copy of the README. And in the main branch, we don't have that. And that's because on my new branch, we have a change that was committed that has not yet been registered in the history of the main branch. So this is kind of heavy for like, I guess, a single cell RNA-seq workshop, but the main way that you interact with GitHub is going to be through pulling, pushing, and making branches that you commit code to. And then eventually you can pull them in to your main branch. So let's say that I wanted to do something experimental on my new branch, and that before I was ready to add it to the main branch where all of my stable software lives, I wanted to make sure it was really working. On my new branch, I would make those changes, I would bug test them, I would do all of the steps necessary to ensure that the changes I had made and the new features I had added were not causing some huge issue with my software. And by the time that I've done all of that and I'm sure that this is working, I can go ahead and commit it to the branch and then finally pull that branch back into the main branch. And in a sense, incorporating all the changes I made back into my main software repository. And this is a way of keeping things separate. So you can test things, you can make fixes, you can make features without having to worry about making some irreparable change to your main software that people are going to yell at you about. And so it's easy enough to pull this change in. You just create a pull request. And then since I own this, I can just merge that pull request, which I'm not going to do since there really is, we didn't add anything useful here. Anyways, long story short, Git is extremely powerful and extremely useful. You should learn how to be functionally proficient in it if you're not already. And for the purposes of this workshop, really all you have to know is that when a change takes place on the remote repository, it's always going to be in the main branch. And all you got to do is come here and do, okay, so let me go back to the main branch, git checkout main. So now I'm back to the main branch. All you got to do to sync up with the remote repository is do git pull. But it's already up to date, so it's not going to make any changes. But if yours isn't up to date, it will pull the new changes in. Okay. All right. So I know this is, again, I know this is a single cell RNA-seq workshop, and like we're not trying to teach you Git here, but in my opinion, one of the things that really is hard when you're first starting out with something like single cell or really any sort of bioinformatics is that there's all these little things that computer scientists know how to do that you don't yet know how to do, and it's not obvious how to do them because you don't have necessarily a CS degree. So this is really for people like that, that I want to show, in essence, how you can do things like working with Git. Okay, so now that we're 30 minutes in, let's go ahead and get started with the lecture. And again, if you want to if you want to know about how to get the data into scRNA from all those different kind of databases and all the different ways that you could find data in the wild, I am happy to go through that later on. Just for the sake of time, I'm going to skip that for right now. All right. So let's go ahead and get started. This module of Bioinformatics Bootcamp is going to be focused on how do you prepare single cell data. So it's going to include things like quality control and normalization and scaling. And we're also going to talk about the upstream components of single cell RNA sequencing analysis. Once again, this is brought to you by Big Bioinformatics. We're a club at UT Health Science Center at San Antonio, and we're if you're also at our university, we're open to new members. If you want to join, just send me an email and I'm happy to send you the calendar invite for our next meeting. All right, let's jump in. So we showed this workflow last time. This is the downstream data processing workflow with scRNA and R. But what happens? Okay, and yeah, and today we're going to talk about quality control and we're going to talk about normalization and scaling. But I also want to touch briefly on what happens before all of this happens. What happens in the upstream component of your single cell RNA-seq workflow? And how do you get from cells to your barcodes, features, and your matrix files? So let's go ahead and take a look at that workflow now. This is the 10x Genomics single cell pipeline. It's the one that I'm going to talk about most because, just to be honest, it's the one that you likely have data from. There are other pipelines, and I'm happy to talk about those. If you have a specific one that you want me to go through, just let me know, and I'm happy to bring it up. But for the purposes of just getting you started and getting you, out of the gate, I'm going to talk about the 10x Genomics one, since again, that's the most popular. So let's first talk about the upstream wet lab side of things. So how do you get to the point where you're ready to start sequencing? Okay, so when you do droplet-based single-cell RNA-seq, and this is really true of not only 10x Genomics, but other methods as well, generally you're going to do something that looks a bit like this. You'll start with beads that have a barcode, an oligonucleotide barcode on them. And so that's right here. This is your beads, and this is the oligonucleotides which coat their surface. Now, these oligonucleotides are going to have an adapter for sequencing. They're going to have a barcode. Now, this is going to tell you exactly which bead the oligonucleotide originated from. So this is how you tell which bead a sequencing read originates from. They're also going to have a UMI that stands for Unique Molecular Identifier. And this is going to be unique for each oligonucleotide. So for every oligonucleotide, this is unique. And then it's going to have a poly-DT capture. All right, so what is the next step? Well, the next step, in at least the case of 10x Genomics, is to run this along with the cells that you want to sequence through a microfluidic channel. So if you haven't had a biomedical engineering course before or a biotech course before, you might not be familiar with what's going on here. Basically, these are very small channels, and you can tell that because you can see the cells in proportion to the size of the channel. While maybe that's not perfectly to scale, essentially we're dealing with something that's probably on the order of around 50 microns, or perhaps 50 to 200. Now you're going to flow water through this first part. So this is all water here. And then you've got an oil phase that's being flown through this part. And this creates a junction right here. This is a water-oil interface. And because of the pressure coming from the water phase, this ends up getting pinched off into bubbles. The idea is that you want to flow together the beads and the cells and ultimately end up with lots of bubbles that contain exactly one bead and one cell. Okay, so that's the ultimate goal. Of course, you will have a lot of bubbles that have only cells or only beads, or they have two cells and one bead, or two beads and one cell, but the ideal situation is where you have one cell and one bead together. All right, so the next step is for essentially time to pass, right? The cells are going to break open and spill their RNA. The RNA is going to be attracted to the poly DT that's on the oligo. So the poly A tail of a mature RNA will hybridize with the poly DT on the oligo. So they'll hybridize together. And then you'll now have a pool of barcoded cDNAs after the reverse transcription occurs. So this, I'm going to go through how that works in just a second. But in essence, so you get kind of the basic idea of it. By the time you remove the oil and you're left with just a big pool of barcoded cDNAs, you're now able to take any of these cDNAs and figure out which bubble and which bead it originated from, because you have the barcode attached to it. And in theory, if you're able to only capture the cDNAs where it was a one bead and one cell situation, then you know exactly which cell it originated from. But of course, we'll see that's not always true. And we'll talk about the quality controls that you use to deal with the cases where you have more than one cell or less than one cell or some other source of error. This can all be taken now and sequenced directly. The other thing that you have is a unique molecular identifier, and we'll talk about that in a minute, why that's important. All right, so this is just viewed another way. This is the gel bead with the oligo. So gel bead here, and this is your oligo here. All right, so the next step, as we talked about, was to add the mRNA. And we can see with the poly A tail, it ends up hybridizing to the poly DT of the oligo. The next step after this is to actually perform reverse transcription, and this happens in the bubble itself. So you reverse transcribe the oligo outwards. So this is cDNA here that's being reverse transcribed outwards, and it's just the inverse or the mirror or the complement, you could say the complement of the original mRNA. All right, so the next step is to attach this TSO, this template switch oligo. And this is really just part of 10x Genomics workflow for library prep. I'm not going to belabor the points that I'm making here about how this is specifically done. I think if you want to learn more about the specific wet lab steps, there are some really great resources. And in fact, you can view this extremely detailed report that will tell you all of the steps that go into this. But for right now, just know that these are sort of the basic first approaches you're going to take. The last part here is to extend the template switch, and now you have the ability to, and now you have what is essentially kind of a mature sort of cDNA here that you can then take and go forward with library prep on. All right, the next step is to actually do your cDNA amplification. And this is because we're talking about individual cells. Really, you don't have enough RNA, and therefore you don't have enough cDNA there unless you amplify it and you get enough so that you can meaningfully sequence it. So you're basically going to use these primers here to continuously amplify your cDNA and create lots of copies. Okay, and that makes it easier to actually sequence it. Now, there's certain biases that are introduced when you do this. So, for example, one cDNA fragment might amplify more than another. Fortunately, you have the UMIs. The UMIs are going to be amplified at the same rate as the rest of the read. And because of that, you can figure out the degree to which any particular read was amplified too much or too little. And that's something that we tend to deal with when we do the quality control pipelines for single cell RNA sequencing. All right, the final sort of steps here all have to do with the actual library preparation. So this is getting ready to sequence. Basically, all you're going to do is get rid of this TSO, attach your adapters for your whatever sequencing protocol you're using. And you also add these P7 and P5 adapters that are for the actual sequencing machine itself. And then the last bit here is the sample index. So this is a 8 nucleotide barcode that tells you what biological sample this originated from. And so if you had, for example, 20 patients, this might be the patient barcode. But this is really just a way that you can differentiate between your samples. All right, so this is the full, this is the full cDNA that is ready for sequencing, right? So when you actually sequence it, you're going to sequence it in paired end mode. So you're going to sequence it both from the left side and the right side here. And when you sequence it from the left side, this is going to be your read one, you're going to get really just the 10x barcode and the UMI sequence. So it's only going to be 28 base pairs long. When you sequence from the right side, you're going to get the actual sequence of the cDNA that is from the mRNA that was captured originally. So this is the actual, when we're talking about RNA-Seq, this is the actual RNA that we're sequencing here. And this is telling you what cell did it originate from and what molecule of RNA did it originate from. So how do we actually deal with this bioinformatically? Well, the pipeline that we typically use is called Cell Ranger. And I'm going to show you in just a second what that looks like because I actually have it on my server running right now. All right, so again, to recap, a FASTQ file is what you'll typically get out of a sequencing machine. And you'll see here, it's in quartet. So I know I talked about this last time, but just to recap, these would be, for example, 3 individual reads on this FASTQ file. And when you look at any individual read, it's got 4 lines. So first line is the read ID and the sequencing info. Second line is the actual sequence. Third line is placeholder. And then the last line is the quality score of each base. All right, so it was just kind of a recap of what we talked about last time, but I thought it would be nice to sort of give a refresher of that information. Okay, so let's now talk about Cell Ranger. So typically when we're talking about Cell Ranger, we're going to be focusing on this very last step. And they've even called it count here, and that's because that's the function in Cell Ranger you'll typically use. And to demo this, I'm going to use the 1000 PBMCs from a healthy donor data set from the 10x Genomics website. So let's just, if you want to follow along with me, you are so more than welcome to do that. But I really suspect you're not going to be able to run this on your personal computer. So unless, like I said last time, unless you're zooming from inside of a high performance computing cluster, this is probably not going to work for you. But in case you want to follow along, I'm putting in the chat right now the page that I'm currently on. So if you want to follow along, you can. So I go to the single cell gene expression data sets. And in this case, I am going to demo the V3 chemistry. So, and in particular, I'm going to do this, this sample here. So I guess I ended up doing the V2 chemistry, my bad, but it's the V2 chemistry in the V3 chemistry dataset folder, which is a bit odd, but there you have it. All right, so what are you going to start from? Well, you're going to start from the FASTQ files. You can copy this directly to the command line. And so this is my server here running. I'm going to go back so you can see this afresh. But let's say I wanted to download this FASTQ files. I just do wget, paste, and I'll go ahead and download it. It takes a while to download, so I'm actually going to cancel this because I've actually already downloaded it. And so there wouldn't really be much point. So I can see this is what it would look like after you downloaded it here. So for those of you who don't know, this is MOBA Xterm. It's the, probably in my opinion, the best client for accessing remote servers that's out there. It's just really nice to have the files on one side and the command line on the other, and you can use the middle click to copy and paste files to the console. So if I wanted to untar this file, I would just type tar xvf, and then I would middle click and it'll copy it to the command line right here. Go ahead and hit enter and it will untar it, which basically just means it's unzipping it if you're used to the Windows nomenclature. Basically, this is an archive, and so we're taking everything out of the archive and putting it into just a normal folder. So it inflated it into this PBMC 1KV2 FASTQs folder, which is right here. And now I can see my FASTQ files right in here. Okay. So for those of you who aren't familiar with Linux or the command line, I would really recommend you try to get that way as soon as possible, because there's so much that you can't do until you are familiar or at least comfortable enough with the command line to try using it. And so if you want, we had last semester, we had a workshop on R and RNA-Seq, and I think week 10 of that workshop and week 11, we showed basic Linux command line and basic Linux environment and how to run commands for bulk RNA-Seq analysis. So the point of this, it's not in the scope of this workshop to teach you Linux, but hopefully this will inspire you to go off and learn it if you haven't already. All right, so let's go ahead and run Cell Ranger now. If I go and I just type in Cell Ranger install or download or whatever you want, the first thing that pops up is the 10x Genomics support page, which is just amazing. It's honestly so good. Their documentation is really nice. But anyways, so they're basically telling you now how you can download it to your Linux server. So basically what I did was I came over here to downloads. I copied this string here, pasted it with the middle click, hit enter, and now it's downloading Cell Ranger onto my computer. But I already have Cell Ranger, so I'm going to cancel with Control-C. And let's just go ahead and delete that. I don't need it. So I already have Cell Ranger. I've already untarred it into this folder here. So that's Cell Ranger right there. So if I actually want to run the Cell Ranger command line utility, it is actually the executable file is this here. So all I have to do is Cell Ranger, tab to autocomplete. So again, if you didn't know that trick, you're welcome. And then tab to autocomplete. So if you just type this in, again, it's an executable file. So just typing it in and hitting enter should attempt to execute it, which it does. All right, so now we have run this program without any arguments whatsoever given to it. So it's basically when you do that, most command line programs will just print back to you kind of a user-friendly usage statement. So it's basically, it's like, okay, you did this because you don't know how to use our tool, so let's tell you how to use it. And that's what they're doing now. They're telling you basically to use this tool, you're going to say Cell Ranger, you're going to give a subcommand, and then you're going to give some arguments for that subcommand. But we haven't specified a subcommand yet. So if I go to the actual tutorial on 10x Genomics website, which is really nice, they walk you through basically how to do what I'm about to show you. But they'll basically tell you in the tutorial what I'm about to say, which is if you're starting from FASTQ files, you can just jump down to the count subcommand, which looks like this. And you can go ahead and run that. And they're showing you an example here in order to align and then quantify the RNA from your single-cell RNA-Seq data set. All right, so let's go ahead and do that now. Cell Ranger, and okay, so again, if you didn't know this, if you hit up, it'll go to the last command you ran. So let's now run count. And all right, this time it gave me an error. It said the following required arguments were not provided, ID and transcriptome. And because it's kind of assuming that I don't know how to use this tool, it's also giving me like a simple usage statement that I could copy and paste and just modify myself if I wanted. And then it's telling me if you want more information, try flag help. So let's try flag help. In pretty much all command line utilities in Linux, if you write this flag help like this and hit enter, it's going to pull up the manual for that command line utility. So go ahead and do that. And you can see here the full list of options you can give to run this. So basically it's saying you should say cell ranger, count, flags, options, and then give ID and transcriptome. So what is ID? ID is the unique ID of a run and output folder name. So it's basically asking you to give the name of the folder you want to output everything to, and also a unique ID that can differentiate this data set from others that you might want to analyze later. And transcriptome, which is the reference genome, along with the known gene locations and transcripts and a few other things that we're not going to build ourselves, but which you have to download from 10x Genomics. And so this is easy to get, actually. They're asking for a 10x compatible transcriptome reference. If you're following their tutorial, they'll literally tell you how to get this. But just for the sake of time, I'm just going to show you where it is. If you go to their website, it's after step 1, it's step 2. So you just go to their downloads page, you find the reference genome that you want. In this case, I'm looking down here, I'm going to take reference 2020A, which is the human genome, GRCH38, which is just another way of saying HG38, which we talked about last time. So this is the latest human reference genome. And to get it and to get all the transcripts that go with it, all you got to do is literally just copy and paste this into the console. But again, I have already done this, so I'm not going to do it again. Go ahead and get rid of you. Okay. Once you untar that, it'll make a folder that looks like this. And if you open this folder, you can see all of the different references that they have in here, references and reference indices that they have. So for example, if you look in the STAR folder, these are all the genome indices that STAR needs in order for you to align RNA-seq reads to the genome. And again, you don't have to generate this yourself, you just download it from 10x Genomics. But it does tell us something about how Cell Ranger works. It basically tells us Cell Ranger at some level is going to want to align our RNA-seq reads to the genome using STAR. You also have these genes, GTF, that includes the, I'll just show you here. So if you look at the head of this file, which basically means look at the first few lines of the file. Whoops, my bad. Clicked on the wrong thing. Basically, what this is going to show you is it's going to show you a tab separated file, that's what this is. It's a tab separated file that includes information about genes, transcripts, and exons. And there's actually, I think, a couple other things that are included here. But basically giving you all the information you can need to know, for example, if you've got an exon, where is that exon in the genome? So it's chromosome 1, and this is the location. It's on the positive strand. It belongs to this gene. And there's even more information here about it. So basically all the information you might need to understand the relationship between the genes and the transcripts and the exons that we know about, and the genome and the locations in the genome. So when you quantify RNA-seq data into exons or transcripts or genes, typically you're going to take a GTF file like this, and you're basically going to use that to figure out, all right, if I've got a read that aligned to this location, what exon did that read align to, for example? All right, so hopefully this, which is probably a criminally rush overview of how references work and how Cell Ranger works, hopefully this is going to still be useful for you, because at the very least, I'm about to show you how you use all of what I've just shown in order to now quantify your FASTQ files, get the count matrix, and then have it in a form that ultimately you can put into scRNA. It's also going to show you how to generate the quality control report. And we're going to talk in a minute about how to interpret that. But first, let's just go ahead and run CellRanger. So go back up here, CellRanger count. So if you recall, we need to add the ID, the transcriptome, and then we also need to give the FASTQ files. There's other options we can specify, and if you're on like a cluster, for example, you can specify that. But I'm not on a cluster, so I'm just going to leave all that as is. So let's go ahead and give it an ID. Let's call it or we'll call it PBMC 1K V2 example. Right? We'll say the transcriptome is in the ref data. So it's in this folder. The FASTQs are in, if you recall, they're right here. So that's our PBMC 1K V2 FASTQs. And that should be it. Okay, so let's go ahead and run this now. Let's see if this works. Okay, now we're running. So believe it or not, this is actually going to take you all the way from the FASTQ files all the way into your count matrix without you having to do anything. And that's because it's not even just one command. It's what we would call a pipeline. And you can see that pipeline if you go here. And I'm going to, if there are anyone who wants to know how to hack my server, I am about to give you the answer. Not really. Don't hack my server. Okay. And if you want to know why I had to just do that, I'm happy to explain. It's kind of outside the scope of what we're doing here today, though. So I'm going to go to my browser to my server's IP address. And I'm going to go to that link that CellRanger gave me. And now we can see the pipeline that is currently running. So this is not like a super user-friendly interface, in my opinion, but at the very least, it gives me some information about the different steps in the pipeline that have to be completed before we can get to the output. So if I come here to the top, basically, this is where we start. And then if I come down here to the bottom, this is where we end. And then we have to go through all these steps in between. But to be honest with you, you're going to actually skip. So these steps are the same even if you're doing different kinds of sequencing. So some of them are going to get skipped because we're not, for example, dealing with VDJ recombination. We're not doing immune cell profiling. So a lot of them are going to be skipped, but a lot of them we're still going to have to do. So if you want to see what step you're currently on, you can just look here. It's the one that's highlighted in blue, and you can get information about it. And you can even see how many resources your pipeline is using by command. And there's a whole lot you can do here. It's not really super important that you understand all this. It's just really nice if you get into the point where you're trying to actually run this yourself to know how to navigate this. But since I've actually already run this pipeline, I'm just going to kill it. And we can now go to the output, which I generated earlier today. All right, so let's go ahead and take a look. This is actually not what I wanted. The output I generated earlier today is in this folder. So when you look at the output, it's going to look like this, right? It's going to have a lot of different random files with weird names. Just ignore all this. What you need is the outs folder. This is where the data you care about is going to live. Okay, so if you are reading these, you'll probably notice that a lot of these file names, you've actually seen things that look like this before. If you come back to the datasets pane in for this dataset, you can actually see that this pretty well mirrors the file names that they gave you here. So where you have, you know, feature cell matrix filtered, you have feature filtered feature barcode matrix. So not perfectly, but this is in essence the same thing as this. And this feature cell matrix HDF5 filtered is the same thing as this. So when you look at the data sets in 10x Genomics website, they're actually really just giving you the output of running CellRanger on the FASTQ files. So what do you do with this data? Well, you could go ahead and start by getting your barcodes, features, and matrix and loading that right into scRNA with the read 10x function. But because this is a quality control lesson, we're going to actually focus first on the web summary.html. So if you're in MOBA Xterm, you can just right click and say open with default program, which in this case is going to be Chrome. So we go ahead and download this from the server and we can open it up. And I guess before we jump into this, because this is going to start us on the path towards the quality control side of things, does anybody have any questions they want to ask about what I've shown here as far as running scRNA goes? Okay, so Wes said, okay, this is a lot of information. Okay. Okay, so Wes made a really good point, which I'd actually never heard of before, which is that the way that scRNA handles your barcodes, the automated way it handles it, might not be correct, and you might end up losing or damaging some barcodes. So this is a really good point. And if you were actually going to all these steps where you're finally now publishing this, you would probably want to go back and redo the alignments, being very careful to make sure that when you run scRNA, you are using logical and correct arguments for it. So if I come back here to my arguments, if I go to help again, I can see here there's a lot of additional information I can supply to scRNA that will allow me to potentially deal with some issues that could arise from maybe inappropriate recognition of barcodes or that sort of thing. In general, I found that scRNA does a really good job. I've never run into that issue myself, but if you find yourself running into that issue, you can always modify the command as much as you need to. And there are a lot of alternatives to scRNA out there. I'm not going to go through any of them in this workshop, but if you want to know about any in particular, I'm happy to go through it. All right, so we're kind of running low on time, so I'm going to jump ahead now to the quality control side of things. So when you finish scRNA, you're going to get this HTML report, and if you open that up in your browser, this is what you'll see. Okay, so what is this telling us? This is a lot of information. The main things that you're typically going to focus on are going to be these panels up here, the plot, and then the percent of reads mapped to the genome. So first of all, this is telling you how many cells scRNA has predicted you actually had in your data set. So again, when we talk about issues in single cell, one of the things that we mentioned before was that, okay, zoom out here. One of the things that we mentioned before was that in a lot of cases, you might have a bead but no cell. So scRNA is actually attempting to predict when this is the case versus when you actually do have a cell and a bead together. And so it's predicted from our data set that there are 998 cells. Okay. So from that, it says that there was a mean of about 77,000 reads per cell, which is actually pretty typical for single cell RNA-seq, and about 1,100 genes per cell. And when it's saying 1,100 genes per cell, what it's basically saying is that there were 1,100 genes that were detected at all that had any reads or any UMIs associated with them. And so that seems like not a lot, and we're about to talk about why that might be when we talk about zero inflation. But just know for now that this is actually pretty spot on to what we typically see with single cell RNA-seq. So how did they arrive at this number 998? Well, to understand that, you have to interpret the barcode rank plot. So you have these situations occurring, and I'm just going to recap one more time. These situations where you have a bead, but you don't have any cells. But what you might actually have in here is some free-floating contaminant, perhaps some contaminating RNAs, Perhaps some RNA leaked out in some earlier experiment and got in here with this. Perhaps there was RNA inside of the media. It's hard to say, but you might have RNAs floating in here. And those RNAs will be barcoded with the barcode from this bead. We can see that situation down here at the end, where you have a barcode. So these are all barcodes along the bottom, and they're ranked by the number of UMIs that were ascribed to that barcode. And so if you look here at the bottom, there are a lot of barcodes that have only one UMI or only 10. And that's really telling us that these barcodes were all basically just capturing noise. Though these are likely to be this situation right here, where you have a barcode and some free-floating RNA, but no real cell is here. And as we go back up the plot and we're getting more and more UMI counts per barcode, we start to have to ask a question of ourselves, which is, where should we make our cutoff? Where should we determine that everything above this point is a real cell and everything below this point is just noise? Where should that be? Well, it turned out that over many experiments that 10x Genomics did, they kept finding this pattern where you would see kind of a smooth arc down and then it would just shoot down like that. And they ultimately decided from some other wet lab validation that they did that the correct place to make their cutoff would be just about here, somewhere in the middle of this curve like this. And this is probably the main way today that we decide what constitutes a cell and what constitutes just background noise. You don't have to follow their recommendation. When you do your analysis in scRNA, you can actually start from the raw feature barcode matrix, which you can load using the read 10x function. And then this will be everything. This will be everything, including the supposed background. So you can set your own cutoff and you can decide for yourself. But I typically will just let Cell Ranger decide, and then I'll typically get more stringent from there. Okay, so the final thing that I tend to look at is the mapping rate. This tells you the percentage of reads which align successfully to the genome. If this rate is really low, that's a really bad sign because that might indicate some kind of contamination. Do note, though, that if you have, for example, a patient-derived xenograph where you have mouse and human RNAs mixed together, this might be low, but you can really explain that based on the fact that it's a PDX. So this is really just more of a rule of thumb. But if you're dealing with something that should just be human data, this should be relatively high. 93% is exceptional. In my opinion, anything above 80% is quite good. Okay, that was a lot of information. Let's actually do some work now. All right, so I want you guys to pull up RStudio, and I want you to pull up the module2.r file. So unfortunately, it looks like we're probably going to run a little bit over time. If you need to leave, that's totally fine, but because you can always watch the recorded lecture later. But if you want to stay and ask questions and get feedback on any issues you're having, do feel free to stick around. And also, if you want to do this interactively with me, feel free to stick around, because we'll probably go just a little bit over time. Okay. Actually, before we even do this, let me just go back here and take any questions. Do you guys have any questions about what I've shown here? Okay, so I got one question that was about multimodal libraries, so ATAC and RNA. Does anyone know how to run RNA-Seq FASTQ files on Cell Ranger without paired ATAC-Seq libraries? I'm honestly not sure, but if you Google that, so if you Google like Cell Ranger, ATAC-Seq, and RNA-Seq, there'll probably be articles that tell you how to manage this. My preference personally would be to run them separately and then put them together in your analysis downstream. So Ollie asked the question, why is the number of genes detected relatively low in single cell RNA-seq? We're going to talk about that. We're going to get to that. The short answer is zero inflation or dropouts, depending on how you want to say it, but the long answer is more complicated. I'll actually show you one thing which I thought was kind of cool. I'm not going to like belabor the point, but basically if you go to the HTML summaries examples folder in the GitHub page, so this is if you download the GitHub, this will be in the folder for module 2. I actually gave two summaries from 2 different single cell experiments that I did. One that did not turn out well and one that turned out well. Basically, this was the one that did not turn out well. This is from a cell-derived xenograft. So this is a cancer cell line that we stuck into mice and we grew it up. And then we did single cell, and this was the result. So it's got an error here. Basically, it says that we had a low fraction of reads in cells. And the reason it's saying that, if you look at the barcode plot, is because, again, this is a log scale. Almost all of the reads ended up being in things that could not be considered cells, right? So based on their cutoff, they said none of this is a cell. And basically that means that most of your reads were occurring because of contamination. Part of the issue here is that this is a very bumpy sort of line, right? You expect that smooth distribution and it goes down quite quickly. We don't see that here. There were other issues with this data set. We tried to process it and it made genuinely no sense. So we resequenced it. And we actually redid the entire, I shouldn't say we resequenced it. We didn't resequence it. We redid the whole process from the tissue all the way down to the sequencing. And we actually did it at a shallow read depth. So we have fewer reads now, but as you'll see, the data looks a lot more correct. So this very nice distribution, we can see here right where it made the cutoff. It makes a lot of sense. And then the fraction of reads in cells is about 72.7%, which is not ideal, but for something that is coming from a whole CDX, which often has a lot of stressed and dying cells in it, this is probably pretty expected. So garbage in, garbage out. If your data looks like this, it's really hard to massage that issue away. So David asked the question, why did we use less read depth? To be frank with you, it's because we wanted to save money. We were trying to troubleshoot the problem with this. And so what we did was we did all of the library prep, and then we just sequenced the library at a really shallow depth. So we only got about 47 million reads in total. And what's nice about this is that now that we know this data is good, and now that we know our library prep is good, we can just resequence the library at a higher depth and then aggregate the data sets together. CellRanger actually has a pipeline for doing that aggregation called CellRanger Agger. It's one of the subcommands that you can read about if you follow their tutorial. So I guess that's one thing I didn't talk about, which might be interesting to mention, is that the sequencing depth that you can continue to sequence at, so the depth that you should sequence at can be easily determined by looking at the sequencing saturation here. And in fact, if you go to the analysis panel, you'll see a nice visualization of the sequencing saturation. And basically what this is telling you is that we could have sequenced at, several times the depth we sequenced. So we could probably sequence all the way up to maybe 500 million or something like that if we really wanted to. And that would allow us to get a more robust data set, and we still would be gaining new information. Basically, once you hit the sequencing saturation, you're really not gaining any new information the more you sequence. So there's a point of diminishing returns, and we really have not even gotten close to it yet. So now that we know our data is good, we can sequence the data at a higher depth, and that allows us to get that robust data set. And the whole point of this is that these are very expensive experiments. You don't want to spend that money if you don't have to until you know that your data is going to turn out well. So now that we know it's going to turn out well, we're willing to spend that money. So that's a trick that 10x Genomics will never tell you. Well, I don't know, maybe they will tell you. I haven't been to enough of their webinars, but it's one that's good to know for sure. Okay, so feel free to look at these reports, and if you have your own single cell data, you can kind of compare yours to these, or you can go to the data sets on 10x Genomics website. If you look at the view summary, basically this is literally the same thing I just showed. It's the HTML report, and you can see this for any of the data sets they have online in their datasets portal. So if you're trying to compare your data set and your quality HTML report to anything that's currently out there publicly, it's easy to do that with these resources. So Wes says that they tell you to do MySeq or NextSeq on low reads before tossing your sample on the NovaSeq. I have not heard that. They were definitely telling us, I think, to do the NovaSeq primarily, although we're used to the HiSeq. Yeah, there've been a few white papers that have come out where they talk about the differences between the different sequencing machines and how well they perform, which are very interesting. But I hadn't heard that they were now telling people to do that. It's very interesting. All right, so let's go ahead and get started. So if you haven't already, go ahead and run lines 4 and 5. These are going to install the packages you might be missing. And then go ahead and load your libraries with lines 8 through 11. We're going to skip part 1 for now. This is where I go through how to load data from different sources. This resource will always be available to you. Okay, so even if you don't stick around for me to go through this today, this will be available, and I'll have a lecture recording that basically shows me going through this. So don't worry about this for right now. Let's move on to all the way down here, part 2. This is line 125, and this is going to be the quality control with single cell RNA-seq datasets. So this is something that I have as kind of a helper line, especially after you finish all of this. You might have a lot of stuff in memory in R, so a lot of stuff in your environment that's taking up a lot of RAM. Every time you need to clear your environment and free up any of the memory that you have on your computer, you can easily do this by running this line. And what it's going to do is it's going to wipe everything from your environment, and then it's going to run the garbage collector, and that's going to immediately knock everything out of memory so that you free up all the memory that you need for anything else that you need to accomplish. So these are very useful for when you're working on like a personal computer and you're running out of RAM. Do be mindful, this will delete everything, absolutely everything that's in your environment. If you need to delete something specific, so let's say I make a new object here and it's now in my environment, so it's now in memory, I can specifically just remove A. So I can just give that to RM and then run the garbage collector, and that will delete it and free up memory. Okay, I'll leave this up here in case anyone wants to see that again later. So let's go ahead and get the PBMC data set. This is the same from last time, so if you don't already have it, just run this and it will download and untar into your current working directory, which is the module 2 folder. And then just like last time, we're going to use the read 10x function to read this into a sparse matrix object. So go ahead and run that. Now we've got our sparse matrix. Then go ahead and run create square root object to make it into a square root object. And we can now see this is our object just like we showed last time. I'm going to zoom in just a little bit here. All right. So now we're going to start examining the QC metrics. And so there are already QC metrics in your PBMC objects because when you create the square root object, it automatically generates 2 of them. So let's go ahead and view those. Those are in the metadata. So how am I doing this? I'm looking at PBMC. I'm seeing the things that are available inside the PBMC object. So someone had asked a question earlier about how to navigate a scRNA object. That's the way. You just click on it in your environment panel, and now you can see everything inside. So if I want to see the metadata, all I have to do is click this scroll here, and it'll show up as a data frame. And this is actually the same thing as just running this view function on line 153. So they both do the exact same thing. Okay, so we already have two quality metrics here. We've got N-count RNA and N-feature RNA. So N-count RNA, that is the UMI counts. So rather than being the read counts, we're counting the UMIs specifically, right? And we do that because you might have had some sort of amplification bias in your reads. And by looking at the UMIs instead of the reads, we can be sure that we are counting the reads that came from a unique molecule of RNA and that we're not counting something that's been over amplified. So this is, other people choose to go with the actual read counts. I've never found an issue with the UMI. There's endless papers that debate the issue. You can look into it if you want, but this is how I personally do it. And it's the default way to do it in scRNA. Okay, so again, if we look at the median of the PBMC metadata N counts, so if we look at the median UMI counts per cell, we can see it's 2197. If we go to our data sets and we look at the, I think this is it here. Oh, sorry, this is the wrong one. All right, so you can get to this is kind of convoluted, so my apologies for this, but if you go to the scRNA PBMC 3K guided tutorial, which is what we're in essence following at the moment, and you go down to where they get their data from, you can see it's right here, right? So if you, I know this is going to sound kind of convoluted, but if you copy this into your browser, and then you change this filtered gene barcode matrices.tar.gz to be, let's see, web summary.html. So if you change this to be web summary.html, it'll pull up the web summary, which sounds kind of convoluted. I'm just going to put this in the chat. It'll probably make this easier. So, but if you want to know about how to find that in the future, that's the way you can do it. So we can see here that in the actual quality report, it reports that the median UMI counts per cell is 2197, and that is what we just saw. The median UMI count is 2197. Median genes per cell. So where is genes per cell here? That's actually this column, N-feature RNA. So that is actually the number of genes detected in that cell. Okay, and then of course our cells are our rows, just to make sure that's clear. So let's go ahead and look at the feature, and we get 817. So just like we saw before, 817. So this just confirms that we are working with the UMI counts in the N-count RNA column and the genes detected in the N-feature RNA column. And if we actually sort these, we can see that there are some outliers in these columns. So for example, these two cells seem to have very high UMI counts, and they also seem to have very high gene numbers of genes detected. So these might be outliers. We might want to remove them. You know, these might also be outliers, but we're not going to do this. We're not going to look at it in this tabular form. We're going to actually plot it to make that determination. There's one more quality metric we want to add in, and that's the mitochondrial percentage. So this is basically asking the question, what percentage of expressed genes are mitochondrial genes? And this is based on the idea that mitochondrial genes are indicative of dead and dying cells, and they're possibly some sort of contaminant. Okay, so you want to remove them because you don't want those dead and dying cells in your data set. So we use the percentage feature set function in order to find, for every cell, the percentage of mitochondrial genes in the genes expressed by that cell. And we can do that with this regular expression here. This basically just says all the genes that start with the line MT dash we're going to calculate the percentage of those. So we add that back in, and now if we look at our metadata, so I should examine that one more time. Now if we look at our metadata, we can see the %MT is also available. Okay, so let's look at the distribution of the UMI counts. So we can see it follows kind of an interesting skewed distribution here with probably some outliers out this way. Let's look at the distribution of genes detected. Again, kind of a skewed distribution out this way. And the %MT, a little bit skewed, but most of it's concentrated low at less than 5%, which is actually a good sign because it means that less than 5% for most cells of the genes were mitochondrial, or the gene expression was mitochondrial. So the way we'll typically actually visualize this, and this is the way that scRNA allows you to do through the built-in function VLN plot, the way we'll typically look at this is with this function here. So if I just run this in the console without assigning it to anything, you can see here it creates a really nice user-friendly plot that shows you the distribution within your data set of the end feature RNA and count RNA and %MT. So it's easy to kind of see where the outliers might be and start thinking about where you might want to make your cutoffs when you filter out unwanted cells. So this is in fact a ggplot object. So hopefully you're all familiar with ggplot. ggplot objects in R can be saved using the ggsave command. And so if I run this ggsave command, I can see that it has created, whoops, there it is. I'm in the wrong folder again, my bad. It has created this qc.png. And this is just a nice high quality representation of those plots. Okay, so the way that we typically actually deal with this is we try to correlate 2 or more of these features together, and then that helps us better visualize the cells that are true outliers that we might want to remove. So we use the feature scatter command from scRNA in order to do that. And I'll just show you here. This is our first plot. So this is pitting end count RNA against %MT. And this allows us to see some outliers out here and some outliers out here. I don't think we've gained a whole lot from comparing these two in particular, but it just does provide a nice way to see where the cutoffs might be made. So we would make our cutoff potentially right here, or perhaps if we wanted to be more stringent, we would make it at 5% so that we preserve the main body of single cell samples or single cells. A more useful plot, in my opinion, is the pitting of the end count RNA against the end feature RNA. And so now we can see that here. We can see where our outliers are occurring. So they're typically occurring above 2,500. And these are the ones we want to remove. And so I'm kind of glossing over an obvious question here, which is, well, how could it possibly be that having more UMI counts or more genes is a bad thing? And the reason is because when we do our statistical analyses in subsequent modules, we're going to be working off of the assumption that our data set has relatively homogeneous technical variance. And we're going to do what we can to make sure that that's the case. But one of the main sources of technical variance in single-cell RNA-seq is going to be right here, the number of genes you detect and the number of UMI counts you have. And if you can just do one filtration step and knock that out right from the beginning, it's really advantageous to do that because it simplifies your ability to manage the technical variance within the rest of your data. So it's something that is quite commonly done. And as long as you're not removing half of your cells, it's typically no one's going to grill you too hard on it. So the way that we actually remove these outliers is, and I'm just going to show this here, we're starting with 2,700 cells, is we use the subset command. So we're saying subset PBMC, and then the actual subset is a logical condition. So we're saying we want to keep the cells that have more than 200 genes expressed, but have less than 2,500. And they also have less than 5% mitochondrial reads. So we do our subset. Let's look at our dimensions again. And now we can see that we've lost about 62 cells. So overall, not much of a price to pay for cleaner data. So I know that we're now out of time. So if you want to stick around, it's up to you. And if you don't, that's fine. But for those of you who do stick around, I'd like you to complete this activity now. So I'm going to give 5 minutes to do that. Basically, you're going to, wherever you see a blank here, that's where there's something you should put in instead. So go ahead and fill in the blanks and see if on this data set, which you can find right here, if on this data set you can get all of the quality control filters that we just created. So I want you to basically generate the violin plots, the feature scatters, and then come up with a subset that you can use to filter the data set. Okay, so go ahead and take 5 minutes, and if you've got any questions, feel free to put them in the chat. So real quick, the DataCamp link, so you should have all now gotten access to DataCamp. If you didn't get that email, let me know, and I'll make sure that it got sent out. It might have also gone to your spam folder, so make sure to check that. You can take any courses you want in DataCamp. It's totally free and open to you. You should have access for 6 months for free. There's no courses that are specific to single-cell RNA-seq as far as I know. Actually, I think there's one, but it uses kind of like an older approach, and I don't know. I would probably not use it for that. But you can also take the, if you're missing any sort of like background knowledge, like on Linux or on like R, or even if you want on Python, you can take courses for that. And there's some really amazing resources for just any area of data science you have an interest in learning about. So abuse it as much as you want. It's totally free and open to you. Okay, so I think that's right about 5 minutes. Before we go on, I just want to pause and ask if anyone was unable to complete that, or has any questions, or is having any issues. Additionally, if you did complete it and you want to present your answer, you feel like you're willing to do that, I would really like that. That way someone can hear, someone else besides me can talk for a bit. It's also fine if not. Okay, so since no one has volunteered, I'm just going to go ahead and go through it. So let's go ahead and run the code. So essentially, this dataset URL, this is giving you the filtered feature barcode matrix, just like we saw previously. So this is your neuron filtered feature barcode matrix.tar.gc. This is the same thing as having gone over here and right-clicked and copied and then just pasted it here. So that's all that is. So how are we going to get this data? We just use download file, and we'll download that dataset URL. And our destination file, it can be whatever you want, but in this case, I prefer to keep the actual name from the original file, so I'm going to do that here. You don't have to do this, you can call it whatever you want, but you could run into issues if you don't at least have the .tar.gc on the end. I also use this method equals curl because one of the issues that I was having on Windows is that the native downloading program, I'm not even sure what it is, was sometimes producing an error in these .tar.gc files. It was somehow adding an extra line or taking away a line, and I found curl wasn't doing that. So that's why I've included this here, and there it goes. Now we're going to untar this. So again, we just downloaded it here. Let's go ahead and untar. And now we've untarred it into this folder right here, filtered feature barcode matrix. So you can see it's got our barcodes, our features, and our matrix.mtx. So all we have to do is point our read 10x at this folder. And I know this is getting confusing because this has probably it looks very similar to the other one. It's not the same, but it's very similar looking. So just keep track of the names when you untar it. Make sure that you know which one it actually is. You can also specify an exdir, and that's a way of saying, hey, I want this to go into this folder, and then you provide a name for it. So I could have provided a specific name, and then it would have been something easier to recognize. So go ahead and run the read 10x. Now we've got our matrix, neurons.mat. And now we're going to create scRNA objects. And do feel free to put in the chat if you have any questions about what I'm showing here, or if there's anything confusing. The odds are that you are not the only person who has that question. So to make this, a really great experience, not only for the people who are here today, but also people who might watch this later. If you have any question and are willing to speak up and ask it, please feel free to do so. It will be a great service to the other people who probably have the same question you have. All right, so we've got our scRNA object now. And now we're going to calculate our %mt. So this is our % feature set. And these autocompletes, again, this is just tabbing. So when you start typing something, you hit tab, you can autocomplete an R. And it's honestly, I think it really will increase the efficiency with which you can do a lot of this sort of work if you aren't already doing this. All right, so now we're going to run our violin plot. And so rather than typing this all out again, I'm going to just copy and paste, because this is not a workshop that's trying to teach you the perfect right way to do things. This is a workshop that's teaching you how in reality you'll probably do it. And I just modify it from PBMC to neurons. All right. I didn't run this line yet. My bad. That would help, wouldn't it? There we go. Okay. So this is actually a much more interesting, from a quality control perspective, a much more interesting and much more realistic data set than the last one. Because you can see, like, for example, percent MT, this is all over the place, right? And you have some that are very high, above 25%. So this is what it's like when you deal with more realistic, real-world sort of tissues. So I think that this particular experiment was from a mouse, an actual mouse brain. So let's see here. So their brain cells. So it looks like it was whole tissue. I actually can't tell from the description here. Anyways, let's do our feature scatter now. So again, don't work harder than you have to. That might be like the golden rule of much of bioinformatics, is that, like, without a good reason, don't work harder than you have to. All right, so now I can see here my N count versus percent MT. So again, this is the UMI counts versus the mitochondrial. We can see that there appears to be a kind of a negative relationship between these. And there also are some pretty clear outliers on the N count RNA. So if I'm looking at this, I might say, all right, there's a lot of cells here above 25%. And that's really just not good. The best that I think we can do to rationalize how we handle this is we can look at the violin plot. We can say, all right, the vast majority of these are, sitting here around 10%. So the upper sort of limit of this bulk group is kind of here around, 15 or 16%. I'm going to set this at 15%. And this is where things become kind of subjective. And it's one of the things that is kind of a drawback about this way of approaching your data, is that at some level, this is partially subjective. Let's go ahead and look now at the feature scatter for the N count versus the N feature. All right, so this is a bit more clear cut now. So we can see here that this goes almost all the way down to 0 on some of these cells, and the bulk of them are kind of in this quadrant here. So if I'm looking at this, the way that I would personally think to subset this is like this. I would say N feature RNA greater than 200, N feature RNA less than, I would probably put this at 6,500. And that will actually also take care of our N count outliers as well, which is nice. So I don't have to deal with N count RNA. So now let's go back to our mitochondrial, and we're going to set percent MT less than 15. So that might have been too conservative. If you don't want to risk losing your initial data and you think you want to relax these later, just start with something that's a little bit more relaxed. And then you can always bump up the stringency. All right, so now we have about 1,100. I forgot to print what we started with. My bad. There it is, 1300. We started with 1300. We now have about 1100. So we've lost almost 200 cells, which is pretty, from just a subjective point of view, that's pretty normal when you're doing your filtering to lose about that many cells in proportion to the total you started with. Some people would argue that you should be more strict or less strict. I prefer to be more strict because I like to have the certainty on a first pass through the data that what I'm seeing is real. So we now have about 1100. Do a more stringent cutoff. Now we're down to about 1042. Okay. So we've now lost somewhere around 250 cells. And at this point, I'm comfortable with the subset that we've created. So I know that in general, quality control is meant to be very rigorous, and in a sense it is, but this part of it is subjective. And once you actually go do the downstream analysis, you might find out that you were either too stringent or not stringent enough, in which case you will always be able to come back here, redo this part, and then run it again. And we'll talk about in the actual biological interpretation part how you can determine whether or not you need it to be more stringent based off of the results you get from something like a UMAP or clustering or a trajectory analysis. But typically you'll find that out when you get to the stage of running your UMAP. Okay. All right, so I really didn't budget my time very well. Again, if you need to leave, you are welcome to do so. This is all going to be online later. But if you want to stay and go through this with me, you're also welcome to do that. So if you need to go, go. I went way over time, and I'm not trying to keep you here later than you can stay. This will be online later. And if you need to leave and you've got like final questions you wanted answered, put them in the chat, and in the lecture video, I'll answer them even if you're not still here when that's going on. Okay. So now we're going to talk about normalization and scaling. And to talk about this, we first have to talk about the problem that this is intended to solve, and why we are not just doing this the same way that you would do it in bulk RNA-seq. So to understand that, you have to understand the character of single-cell RNA-seq data. So as we mentioned before, and you can see this if you look at neurons.mat, most of your counts are going to be 0. Most of your data set is going to be 0. Oh, someone asked a question. They're seeing this warning suppressing 32 column names. This is not a warning. This is actually just a message. It's basically just telling you that there are column names in this matrix, and we're just not showing them to you because we don't have the space. So that's all this is really telling you. It's really, you can really disregard this. All right, so this is sparse data. That's what we call this, sparse data. So what is the character of sparse data? Well, let's go back to the PowerPoint now. So I'm jumping back forward to where we started. Okay. All right, so I call this the sparsity challenge, and it is actually considered a serious problem facing single-cell RNA sequencing. There is a fantastic paper that came out not too long ago called 11 Grand Challenges in Single-Cell Data Science. So if you want to dig further into the theory and sort of the debate behind how we approach single-cell, this is a fantastic place to start because it outlines the 11 biggest problems facing single-cell today. And what is problem number 1? Handling sparsity in single-cell RNA-seq. That is the very first challenge, and you can see how important they see it as, considering they put it first. So basically what they're describing here is a problem that I'm about to demonstrate. When you look at a count matrix from single-cell RNA-seq, it looks something like this. I've pulled up part of a matrix in Excel to demonstrated. If we look at a particular row, we'll see that in some cases you have almost all zeros, but you might have a 1 or a 9. And so let's go ahead and color this by the value. So if we wanted to understand, for example, how is this gene IGS-15 differentially expressed between cells? Well, it seems pretty straightforward. Clearly, it's overexpressed in this cell and partially overexpressed in these cells, and it's not expressed at all, or it's underexpressed. We might say underexpressed in these other cells here. But is that actually a true representation of the biology? To be certain that this gene is actually being overexpressed, we need to first have a certainty that when it says 0, that it actually is 0. So this brings us to the 3 points about uncertainty in single-cell RNA-seq. The first point is, how do you know if it's really 0? Second is, how do you model this data when most of the values are 0? So what stats can you use? And then and point 3 is really, how sure can you be about your analysis with so much statistical uncertainty? So let's address point 1 first. The other points we're actually going to get to in the course of talking about how scRNA works. But point 1, we need to talk about upfront. This comes from a really fantastic single-cell RNA-seq workshop that is available online. So if you go to this link here, you can see it. They developed this visualization, and rather than recreate the whole thing, because it was so good, I decided to just use it here. So when we talk about the initial RNA population that you could be sampling with single-cell RNA sequencing, we often talk about dropouts. And what does that mean? Well, that essentially means that perhaps when you did your reverse transcription, when you did all the wet lab prep steps we talked about earlier, there was some gene or some transcript that for one reason or another did not get captured on the bead, and therefore it never got reverse transcribed, and therefore it never got amplified. And so then when you sequence it, even though this gene and this RNA was being expressed, it is not going to be present in your final data set. It's going to be 0. And you will not know in your final data set, you will not have any way to tell whether or not that 0 means that the gene wasn't expressed, or the 0 means that it simply didn't get amplified correctly. This is what we would consider to be the ground truth. If we could know the ground truth, this is what it would kind of look like. You've got cells and you've got genes, okay? So basically this is showing the expression in each of these cells of some particular set of genes. So some cells are going to express these quite highly, some cells are going to express these quite highly, and this is what you could use. If you could know this truth, you could use this to very robustly analyze your single cell data. You'd be able to tell exactly where the clusters were and where the trajectories are occurring. In reality, it looks like this. So you have lots of missing values. You have lots of places where you should see some signal, but you don't. And so how do you deal with this? This is the phenomenon we refer to as dropout, or zero inflation is kind of the other way that we talk about this. Although people will argue you shouldn't use these terms interchangeably, they are used interchangeably in the context of most papers. So how do we handle this? Well, the first way we can handle it is by using a statistical framework which is robust against zero inflation. And that is what scRNA provides. The second way is to impute the missing UMI count values. So what does that mean? Let's talk about this since we're not actually going to do it during this module. I'm going to show you an example of a really popular technique called magic, which is used for doing this. And essentially, you can see it down here. I'm going to zoom in a second. Essentially, you take this sparse data set, which is full of zeros. You figure out which cells are most similar to each other. So you construct a nearest neighbor graph, which I'm going to explain next time. But for right now, this basically means that, for example, if you were trying to predict my nationality, okay? So you're trying to predict my nationality. You are able to tell who my 5 nearest neighbors are. So my literal, like I'm in my apartment right now, the people who live closest to me, the 5 people who live closest to me, those are my 5 nearest neighbors. Let's say you don't know my nationality, but you know their nationalities. You could impute my nationality by saying, well, okay, 4 of them are Americans and 1 is, an Indian grad student who's, here in the same PhD program. So you could say, most likely, I am an American as well, because 4 out of the 5 were American. But I might not be. So that's the problem. You can see what the limitation here is. Just because your neighbors have something doesn't necessarily mean you have it. And so there's actually quite a bit of criticism of this approach. I use this sparingly, and when I use it, it's only on data sets that I feel like have the depth of sequencing necessary for being able to confidently make that imputation. But there are entire dissertations written about when to apply imputation and how to apply it and what tools to use. It's not what we're going to be using here, but because it's such a popular approach, I wanted to make sure I talked about it briefly. All right. So let's actually jump back in and see how we do this in scRNA. So the first step in scRNA is to normalize the data, and all they're actually doing here is they're log transforming the counts. So this is whatever the count was, you add 1 to it, and then you take the log of that. And that's all done with this function normalize data. So we run that. Let's drop this back. All right, and now we've got our log transform counts. The next step is to find variable features. This is a very math intensive and honestly pretty complicated step, and I'm not going to explain how it works here. If you want to know how it works, go read the scRNA papers, and also I would read about their SC transform approach, because that kind of explains the intuition behind a lot of this. You can also get an idea of the intuition for how this is working by looking at the help page for this function. So if we come here and we look at the help page, we basically are going to, so we can come down here, read this. VST is the default method, and it's going to explain how this is going to do this. So you can read this, you can read the papers that this comes from, and you can dig as far as you want into the math. I'm not going to do that here because it is way beyond the scope of what I want to show. Just know that the next step in this process is to run this command. Okay, so variable features are the genes that differ greatly in expression between cells in your sample. So these are, if you've ever done any sort of unsupervised machine learning, these would be your highly variable features. So let's go ahead, we're going to find the top 10, and if we print that to the screen, we can see the genes that are the most variable in our data set. And then we can also make a plot where we label these. So this is kind of a nice way to represent this. It just takes a second. Okay. So this plot is going to pit average expression for every gene against the standardized variance across all cells for that gene. So basically what this is telling us is that PPBP has the highest variance, which is essentially a way of saying the expression of PPBP differs greatly across all the cells in your entire data set, which means this is probably a strong marker for determining how some cells in your population are different from others. And it's one of the things that we're going to use to ultimately do our principal component analysis, our UMAP, our clustering. And in fact, all of our top 2,000 variable genes we'll use for those purposes. All right, so The next step is to scale the data. And scaling a data set is really just a way to make the mean 0 and the standard deviation 1. So that's in general what scaling means. I think they have some slight alteration on that in this context, but in essence, that's what we're doing here. And the point of that is that when you want to do anything like principal component analysis, you have to have centered and scaled data. Okay, so centered meaning that the mean is 0. So there we go. Now we have centered and scaled the data matrix. The other important thing to consider here is that we had to first calculate our variable features in order to do this, because what we're essentially going to do is we're going to take the highly variable features and then scale those. We're not going to scale everything in the data set. And the reason is because if you have a lot of genes that are only 0 across all samples, one, they're not going to be in your list of highly variable genes, so we're not going to consider them anymore for our downstream analysis. So if you attempted to scale data that had a bunch of zeros in it, it would be very skewed, right? So now that we only have the highly variable features, we're able to scale those, make the mean 0, and then easily see how those features are contributing to differences in the data set when we do our principal component analysis, and then downstream from there, all the other things we'll be doing. Anyways, if you want to actually see what the effect of these various scaling approaches that we just did, what the effect of that is, you can run this line 240. It's kind of large and complicated. But in essence, what I did here was I basically created a plot that shows the distributions with the raw counts, the log transform counts, and the scale counts. So go ahead and run that, and you can now see that here. Let's zoom in on that. Okay, so in our raw counts, we can see that almost all of them are 0. So this is a box plot, by the way, and yet you can't even see the box because everything's basically right down here at 0. You can only see the outliers, which are the ones that have any real counts. Once we log transform it, all of our 0 counts are still 0, but we can now see the non-zero counts are better spread out and they're better distributed. So it's a bit easier to compare between our two cells. So these are, I guess I should mention, these are 2 cells here. Sorry if that was not clear at first. I only picked 2 cells to compare this on. And now we can look at our scale data set. So again, these are only 2,000. These are the 2,000 highly variable genes. But as you can see here, it's a much more, I guess you could say, approachable from like an unsupervised machine learning perspective or PCA perspective. It's a much more approachable data set, where now, since these are highly variable genes, if you've got a gene that's varying between these 2 conditions, say it's overexpressed here and underexpressed here, you'll be able to easily see the differences between them, or more easily see the differences between them. This is obviously not something you would do by hand. I'm really just showing this for illustrative purposes, but it's really just nice to be able to see what the impact of these various transformations you're making are on the data. Okay. So I'm going to give another 5 minutes. Go ahead and do those steps on your neurons data set. You don't need to do this last one unless you really want to. But the rest of these, the normalized data, the fine variable features, and the scale data, go ahead and run those. I'm going to give 5 minutes for that. And then we'll wrap up. And if you have any questions, you run into any issues, do feel free to put those in the chat. Okay, so let's go ahead and jump back in now. So hopefully you took my lesson from last time to heart, and you learned how to use the laziness inherent in all of us to become a more efficient bioinformatician. All right, copy and paste the whole thing. And if you didn't know how to do this, you're about to learn. Find PBMC, replace with neurons. Replace all. And there you have it. All right. So learn to be efficient by embracing your inner laziness. As long as you're not making a mistake, which try not to make those, there's really no reason not to do this. Okay, so go ahead and normalize neurons, find variable features, get the top variable features, plot. Whoops, my bad. Looks like I didn't give it enough space. That's fine. All right, and now you're seeing the top variable features. Some really interesting stuff in here. Bimentin, top 2A, ID3. So some clear kind of mesodermal, maybe sort of stuff going on here. Top 2A is kind of interesting from like a cell cycle and whatnot perspective. So there's a lot of interesting stuff in here, I feel. What's more important for right now, because we'll really get to the interpretation later, what's more important for right now is that you've got your top variable genes and you can now scale your data. There you have it. And that's all it takes. All right, so I was going to have us go on to PCA. And in fact, I'll have us do that. I'll explain how PCA works next time, but just know for right now, all you got to do is hit this run PCA command and it'll just do the whole thing. So run PCA. And it's done. And now you can plot your PCA. And this is your PCA. And this is where I'm going to leave it for right now. So as promised, when I thought I was going to be done sooner, I'm going to now go back and show you how to get the data from different kinds of sources. If you wanted to listen to me tell you this, I am happy to have you stay on the call. If you want to go, you're welcome to do that if this is not interesting to you at this point. But for right now, I think that we're going to go through this relatively quick. And then if you want to watch this later on the recording, feel free to do that. All right, so let's go ahead and do this. So the first way to get data is the easiest and most impractical of all, which is to get it from the 10x Genomics website. So this is, again, I'm in part 1 right here, starting on line 13. So to get it from the 10x Genomics website. And so, for example, I showed this last time. You can get the data by going to the data sets, and that's how you can get all the public data sets that are out there. And, you know, browse to your heart's content. They're pretty easy to actually get. So in this case, we just downloaded the filtered feature barcode matrix.tar.gz from this PBMC 1KV3 experiment. And this we've already shown. So like we showed last time, you just run this and then it goes through like normal and you can make your square out object. Very easy. The read 10x function works perfectly for this. So let's say instead of the, well, actually, before we go too much farther, let's clear the environment because we don't really need anything in the environment for this. All right, so we're going to free up some memory. All right, so now let's, instead of using the filter feature barcode matrix.tar.gz, we're going to use the H5 file. So as you recall from our Cell Ranger pipeline, it generates the filtered feature barcode matrix in an H5 format as well. This is a really, really useful archive format for dealing with large data sets that have multiple components to them. In this case, you can actually use a built-in swear out function in order to read it in to a matrix and then create your swear out object. So In this particular case, we're just going to download it using a variation of the command from last time. All right, my R session is starting to slow down on me. All right, I'm gonna refresh my R session in 1 second. This is something you might want to do periodically because R will over time slow down for reasons I don't entirely understand. The longer you use it or the more of something you try to do with it, it might slow down. All right. So now we've got our PBMC filter gene barcode matrices .h5. And we can use the read 10x h5 command to read this in. So very easy to do. And this all works with data that you generated using the cell ranger pipeline. Let's talk about other data sets and other databases that are out there. So PangLauDB is a really fantastic resource. Basically, if you check it out here, if you go to the, actually, let's just go to their home so you can see what's going on first before we start downloading. So basically, they're a resource for reprocessed and standardized single cell data sets. And the really nice thing about this is that it's very easy to get started with one of these. So let's start looking for a data set, okay? I think I chose, for this one, I chose 553822. Okay, so that's this very first one. Whoops, that's not what I meant to do. View. And so you can see the data page for it. But what we really want is the read counts in the R data format. Okay, so you can actually just right click on this. You can, oh, actually, yeah, I'm not sure if this works. Now that I'm thinking about it, I don't think even the curl method worked for this one. So you can just download it directly by clicking on it. The issue is that they made it such that they don't want you programmatically accessing it with like download.file. So it's really, I'm pretty sure you can only do this through your web browser at this point. There's probably a way, I'm just not sure how. But anyway, so now you've got their R data. You can now transfer it over to your working directory, paste that. And you can just go ahead and load it up. In this case, it appended like a weird 1 to it, probably because I downloaded it previously. So I can modify this to be the correct file name. And then to actually load this into R, all you have to do is use the load command on that file. So this is an R data file, and it contains environment objects that can be directly loaded into R. In this case, all that was loaded was this SM. Okay, so that's what got loaded. It's a DGC matrix, which is great. And we can directly create our square out object from it. So simple as that. PangLauDB is a really nice way to get access to a lot more data sets without having to reprocess anything yourself. All right, so let's now go to the main way that you'll be getting your data, the most practical way and the hardest way. So I'm going to actually show you from a study that I managed the accession on in Geo, where you can get some single cell data. So for those of you who don't know or who haven't used Geo before, just do a quick Google search for Geo. Ignore all the stuff from this company because it's actually, that's a different thing. And come down here to the NCBI website. So I'll put this in the chat in case anyone wants to follow along here. This is the main repository for all publicly available sequencing experiments. And the really nice thing about Geo is that because for high impact publications, and really publications in any reputable journal at this point, you have to make your data publicly available. Almost every high throughput sequencing experiment that's been published in the last 10 years, you can get the data through this portal. And the sister portal, which is called SRA. So I come down here to search Geo datasets, and let's say I'm trying to mine some public data set related to Ewing sarcoma. So I would type Ewing sarcoma single cell. So that might be how I would type this in. And I'm going to zoom in a little bit here so it's easier to see. It'll present you a big page of results. So you can easily filter your results to include only the data type you want. So in our case, we're looking through genome, not that one, expression profiling by high throughput sequencing. That's the one we're looking for. So we can apply that filter. And we can also, if we had other species here, we could also select only Homo sapiens if we wanted to. There's other filters we can apply. But now that we only have 9 items, it's pretty easy to look through this by hand. So as you can see, I've already clicked on this first one, and as you could probably guess, that's the one we're going to be using. So this is actually an accession that I manage. This is the first page that's opened up here is the series accession page. So the accession is this code right here. It's right here. It's what you have to put in your publication, and it's what allows other people to find your data. And then when you actually come to the series page, which that's what this is, another way to think of this is the page for that study. You can read the summary of the study. You can read about the papers that were generated as a result of the study. So these are the papers, actually these are the same paper. For some reason they've been listed twice. This is the paper I wrote. And you can also see the samples that are included in that study. So we've got 3 cell lines, CHLA9, CHLA10, and TC71. So if I click on one of these, I'm now going to be taken to the sample accession. So this is the series or the study. This is the sample accession. So it identifies a unique sample within a series. Now this is going to describe how this cell line was prepared, how the sequencing was done, and it's going to also describe how the alignment was performed. It says HG38, that's the genome it was aligned to. It was Illumina NovaSeq 6000. And we've got our barcodes, our features, and our matrix down here, which you can download. Okay. Now let's say that I want to mine this whole study. Okay. The way that I can easily do that is by downloading this supplemental file, this GSC raw.tar. So what does this file contain? Well, it contains all the files that are within the sample accessions. And so if I click on custom, I can see all the things it contains. It contains the barcodes, the features, and the matrix for each of my 3 cell types. Okay, so easy enough to download this through R. If we come back here, we can actually just do a sort of simple download.file here. So I've given the archive URL, which is if you right click on, if I click cancel here, if I right click on this and do copy link address, that is what this is. So I just pasted that, it's the same. Go ahead and download that. We use the curl method here because when I tried to do this with the default method through Windows, it ended up having a corruption issue. I think it appended something to the files or it deleted something from them, but this is the way that I found to work on all environments I've tested so far. All right, so it's downloading. It's a little slow because it's a lot of data, but it's always going to be a lot of data when you do this. So just give it a second. All right, and there we go. That should be about, if you hit refresh, you can see the size of the file increasing. Okay, now it's done. Now we're going to untar it, and I'm actually going to specify the directory as being called EWS cells 10x. All right, so now I've got the directory, and it contains all of the barcodes, features, and matrix for each of my cell lines. So this is clearly 10x genomics data. You've got your barcodes, features, and matrix. Can you just use read 10x on this data directory? No, you can't. The reason is because read 10x only works when you have a situation like this, where you have a directory that only contains the files, barcodes, features, and matrix, and they're named correctly as barcodes.tsv.gz, features.tsv.gz, and matrix.mtx.gz. So because we don't have that, we have to do things in a little bit more DIY kind of way. So let's say we want to start by just processing TC71. We're going to first read in the matrix file for TC71 using the readmm function from the matrix package. So this is actually what scRNA is using under the hood, but now we're going to explicitly call it so that we can get this all into the correct format. So that is this file right here. So go ahead, read that in. And now we've got our DGT matrix. So we're used to seeing a DGC matrix. We're now seeing a DGT matrix. Why are these different? Well, the way you'll handle them is not different. The way that they represent data and the way they compress the data to represent it is different. DGT matrices have a triplet form, and DGC matrices have a compressed column form. But the way you actually handle the matrices yourself will probably not ever change, because ultimately all scRNA can handle this as well as a DGC matrix. So don't really have to worry about that. So now we can get the genes from the features.tsv.gz, and we'll use the read tsv command to get that. I set column names equal to false because there are no column names in this file. If we read this in, we can see here that the first line, it starts right into the genes, right? There's no column names. If there were column names, the first line would be the column names. Okay, but there's not. So it gives us X1, X2, X3 as kind of a stand-in for where column names should have been. And this is going to give you all of the genes, and this is basically the rows of your matrix. So it's very easy for me now to assign the genes and the gene symbols. I'm using the gene symbols rather than the ensemble gene IDs to assign these to the row names of the matrix. So if I look at my matrix just by itself, there's no row names, it's just numbers here. If I assign now the genes to the row names, I can see that it now has the genes as the row names. But it's still missing column names. There's no column names. We're not seeing any warning like, oh, we're hiding the column names from you. That's because there's no column names. And if you type in column names, you know, TC71 mat, it says null because there's no column names yet. So let's add them in. They should be the barcodes that we're trying to use for this experiment. So we go ahead and read in the barcodes.tsv.gz. And again, call names equals false. It gives us this one column matrix of the barcodes, or sorry, one column tibble of the barcodes. And we can set those as the column names for the matrix. Okay, so now if we print the matrix, just the first few lines and columns, we can see it's now got the genes and the column names. And this can be fed directly into scRNA to create our objects. So let's say we wanted to do this again, but we wanted to do it for CHLA9. Okay, well, that's easy enough to do. We're basically just going to repeat what we just did for CHLA9. So that's lines 75 through 82. So you can just go ahead and run those. Okay, and now we've got our scRNA object for CHLA9 as well. So if we print them, scRNA CHLA9 and scRNA TC71, we can see how many cells are in each, how many features. It's the same number of features, so that's actually not an issue, but the number of cells differs. Now, one thing that I did here that I hadn't shown in the past was when I created the scRNA object, I gave a project identification. And so I called this one TC71 and called this one CHLA9. So where is that stored? Well, if we look at, I'll just show you here how to do this. If you look at the SRT TC71, again, where is all that stored? Well, it's in the metadata. And we can see here now that orig ident is a factor with one level TC71. And this basically tells us that rather than being called like before, I think before it was just called scRNA project or scRNA dataset or something like that. Now we've actually given it a real name that identifies this scRNA object as TC71. Why is this important? Well, because now we're going to merge these scRNA objects into one combined object. So we've got X is scRNA CHLA9, Y is SRT TC71, and you just merge them like that. It might complain that some barcodes were duplicated. That's totally normal. It'll rename them to make sure that they're unique. You don't have to worry about that. So now if we look at this object with our combined data, we see that it has now 13,000 something cells. And if we do a table on the original ident, we can see that it has preserved our CHLA9s and our TC71s. And even though they're now merged together in one object, we can clearly identify which is which. All right, I was going to have you do this activity where you add CHLA10 in as well, but for the sake of time, I'm just not going to do that. Essentially, all you would need to do is do the same thing for CHLA10, and then you could combine them just like we showed before with X and Y, X being the CHLA9 and TC71, Y being the SRT CHLA10. Final way to look at this is, we're doing an awful lot of copying and pasting here. There's probably a more efficient way. And in fact, there is. So this is an lapply that I came up with to basically create this as a list. So the way this is going to work is it's going to loop through CHLA9, CHLA10, and TC71. For each one, it's going to find the matrix, it's going to find the features, and it's going to find the barcodes. And then it's going to basically read in the matrix, read in the features, read in the barcodes, and then create the scRNA object with the name. So whichever one of these is currently active in the loop, and then it's going to return it. So what does that look like? Well, this might take a second to complete, but I think it's important to see how this is working. One thing I changed when I did this is that instead of, up here, I did this genes and I did the $sign to access the X2 column. Because it's a tibble, I can actually just use the dplyr pull verb in order to pull that column out as a vector. So that's just a nice alternative way to handle it. All right, so it's looping through. The nice thing about this is that no matter how many additional cells I add or samples I add, this will still work, as long as they're all still in this folder. So let's take a look at this SRT list. So we can see now it is a list comprising 3 scRNA objects. If we look at the first one, we can see it's the one that's got 5,077 samples. If we look at the orig identity, we can see that's our CHLA9. And we would find that if we want to go through each of them, this would be CHLA9, I think this is CHLA10, and then TC71 right here. But the point is to combine them. So here's a trick you can use. SRT list 1, so the first element of SRT list is your X. And you're basically saying here, make my X the first element of this, which is this object right here. But your Y can actually be a list. So we're basically now saying the SRT list with everything except the first entry. So we're basically saying a list containing both of these last two. We're saying combine these together. So go ahead and run that. The nice thing about this is that this will work no matter how large SRT list is. It will merge all of the scRNA objects together from that list, no matter how large it is. So this is a really great trick to learn. And now we've got full SRT, which was created, and it's got 18,000 something cells. And if you look at the original identities, we can see, let's table that. we can see here it's got the correct numbers of CHLA10, CHLA9, and TC71. So if I went on, for example, to do my quality control, I could separate my violin plots by identity. And in fact, for demonstration purposes, I think it's probably instructive to see how you would do that, just to see what the sort of logical outcome of this would be. So I'm going to go ahead and, just like I taught you, copy and paste. So I'm going to go ahead and do that. So wherever it says PBMC, you know what to do. Find and replace PBMC with full SRT. Do not do more work than you need to. All right, so we got the percent mitochondrial reads. I don't really care about any of this. And I really just want to see the violin plot now. So let's go ahead and look at the violin plot. This might look a little bit messy at first, but just bear with me. So this is the violin plot with everything, and we're not going to differentiate at all based on the cell line. Okay, so ton of data, way too much to easily interpret. Oh, actually, no, we did differentiate it. I guess by default now it does stratify them. Okay, great. Actually, that does make sense because the identity was their cell line. So this is perfect. Okay, it's going to take a second to adjust, but all right. So hopefully, what you're starting to be able to see here is we can now compare our 3 cell lines, okay, with respect to end feature RNA, end count RNA, and percent MT. So what is kind of nice to see is that there is a lot of genes being expressed in our CHLA10 and CHLA9. They're cancers, so that's kind of expected. There's a lot of UMI counts, which is great because we want a really robust data set. And there's pretty low percent MT. TC71 is another story entirely. This is actually, I didn't mention this before, but this is one of the ones that failed earlier. So we didn't use it in the paper, and I think we mentioned this in the paper that this one failed. We included it just because we want to give people access to our data, even if we don't use it ourselves. We really just use these 2 in the paper. But essentially what you're seeing here is that if you want to use this data, you have to contend with the fact that almost all of it has either high levels of mitochondria, low levels of UMI counts, or low numbers of genes expressed. So this is another Ewing sarcoma cell line, just like these 2 are. So really, it shouldn't be that terribly different. And in fact, when we redid the data later in our new run through Cell Ranger, it actually turned out fine. All right, and I mean, in future papers, that will get made available. We just didn't have that at the time we did this. Okay. So that's in essence how you can show your quality control features, stratify by cell type, and then make cutoffs specific to each cell type. So for example, if I wanted to subset this, what I would probably do is I would probably set my cutoff, here and here, and then I'd probably set it here and here, and then I'd probably set it here and here, and it's easy enough to do that with the subset feature. I actually don't see any need to subset specifically by cell type here, so I probably wouldn't, but you could if you wanted to. And I'll show you, just for the sake of you knowing how to do that, I'm going to show you how you would do that. So let's say that I wanted to apply a rule. And I'm going to scale this back here. Let's say I wanted to apply a rule only to TC71. So the way that I could do that logically, let's say I want percent MT less than 5 to only apply to TC71. Okay. All right. So the way that would work is the column is, and let me just fix this. Oops. The column is orig ident. So we could say here, oops, and okay, let me think about this. Right, okay. So we could do it like this. So we could say, all right, come on. Orig ident does not equal TC71. So this is what I want to be applied to everything that's not TC71. Okay. So either your cell is going to be one of these two and it's going to have these things true. Okay. Or, and that's how you say or in R, or orig.ident is equal to TC71. And I think I've gotten a little bit messy here, but and percent MT is greater than 5 or less than 5. That's what we said. So that would be how you could do a subset where you incorporate the information about which cell type it is. Okay, I think that would work. Yeah, that seems to have worked just fine. That probably really reduced my data set. Yeah, by a lot. So yeah. All right. Yeah, that's because most of the end feature RNA for these two was definitely above 2,500. So that was probably a pretty bad cutoff to choose. But you know what? I could always go back and redo it later, just starting from right here. So if I was actually doing this, just to give one final example, I would probably choose a cutoff of, 8,500. I'd probably choose a bottom cutoff of, 4,500. I would probably choose a mitochondrial cutoff of less than 15%. And so we got to keep a lot more of our cells that time. And you can see we still have it correctly stratified by cell type identity. All right, so I guess the final example, which, if you want to see it, if you're still here, it means I guess you want to see more. I'm going to show one final thing, which is what if all you have is like someone in your lab left and they had a bunch of files in a folder and one of the files said single cell RNA-Seq .tsv, and like you open it up and it looks like a read count matrix. So let's say, what about the situation where all you have is a TSV? Okay, so that's actually pretty easy to do. So to actually simulate that, I'm just going to write I'm going to write this PBMC data to a TSV. Hopefully that goes real well. Write that TSV. So I'm going to convert it to a data frame, PBMC df. Right, because I cleared that from my environment. Okay. Let's read that back in. All right, so now we're converting it from a matrix to a data frame using the as.data.frame method, and you can see it right here, PBMC df. I would not recommend opening this, as RStudio will not enjoy that. It will have a really hard time displaying that meaningfully. Okay, so let's say we want to write that to a TSV now. All right, and let's just call this scRNA-Seq.tsv. This is like trying to simulate something you might actually encounter in the wild from like a lab mate or from like a PI who gives you inherited data. Okay, so we're going to write that to our TSV. And let's go ahead and see that here. It takes a minute to finish, but now it's done. Okay. So let's pretend this is what you get. You've now been handed a TSV file, and let's say that you go into, this is, I think, a common way to try to deal with this is to like try to open it in Excel. which apparently I don't have this set up to do yet, but you can kind of get an idea of what it is by just looking at the notepad or WordPad or whatever text editor you want. So you can see you've got barcodes, and then you've got a bunch of zeros. Okay. And actually, I made the mistake of dropping the row names. So, all right, so the row names are supposed to be there. So we're going to write it with the row names. It's my bad. Oops. This would be the write.table function. When you do it with dplyrs, or sorry, with readrs write underscore tsv, it'll actually get rid of the row names, which is what just happened. Row names equals true. Separate. If you want to write a tsv file using the normal built-in R function, you just do this write.table, and then you make the separator a tab, and that's what makes it a tsv. All right, so it's going to take a second to write that. Sorry about that. Shouldn't take too crazy long though. All right, but going back to what we saw before, when you do encounter this in, as I say, in the wild, do feel free to also tell your boss or your lab mates or whoever that you really like to see the source for this data, because you really have no idea how this was created or how the upstream processing was done. And without that documentation, you might fall into some pretty bad traps as far as how you deal with the data in a quality control context. Yeah, write.table is a lot slower than write TSV, unfortunately, so it's a bit slow. Okay, so this will probably just take another minute. So for the people who are still here, do you guys have any questions that you wanted addressed beyond what I've kind of already talked about here? I know we've really gone through a lot, but I want to give you all the information that you need to be able to do this on your own. So do you guys have any other questions or anything else you want me to talk about? Feel free to put that in the chat while I read this in. All right, so I've now created this TSV. I'm going to open this with Notepad. So like before, we've got our barcodes, and now we've also got our row names here. So our row names are also included. So let's say that this is what you inherit. Inherited .tsv. This workflow is the same if you inherit a CSV. The only difference being that instead of using the read.table command, you would use the read.csv command. All right, so let's read this in to R, and we can actually use, to speed up the process, we'll use read TSV. Okay. So we'll call this scRNA counts. All right, and we can already see that there's a mistake, which is that it has considered only one column, so it thinks that there's only one column here for some reason. And so this is where you'll run into just various issues and challenges working with this sort of thing. But it seems to have actually corrected the issue, so that's nice. So if we look at scRNA-Seq counts, we can see that even though it gave us a warning and it basically said it only found one column, that it actually was able to figure out the other ones as well, which is honestly kind of funny that it can do that. The main issue that you now have, which is very clearly displayed here, is that the barcodes were all shifted over by one. Okay, so that's why when you're doing this, one of the things that you can do is to, when you're either making the table in the first place or when you're reading it in, to assign a a row name column. So I don't think that read underscore TSV lets you do this. You can finagle it in other ways, like you can shift everything over in other ways, but for the sake of time and simplicity, I'm just going to use the read dot table for this. And I'll set the sep equal to t. So let's do it that way. That'll just make it more simple for this demonstration. And let's actually set the row names equal to 1. So what is that doing? Well, that's basically saying the first column is the row names. So when you build the data frame by reading this in, set the first column as the row names. All right, now it's done. I would really recommend not printing what I just made to the screen, because that can end up making R very unhappy, because it's a data frame, not a tibble. When we printed it before, it was a tibble because we used read underscore TSV. Now it's a data frame because we use read dot table. Data frames do not print as cleanly, but we can see that it is correct. We've got our barcodes, we've got our gene names, and now all we actually have to do, as crazy as this sounds, all you actually have to do now is run create scRNA. And there it is. In a weird way, even though this is like the most basic of all possible formats for your data to be in, this is actually the easiest one to just straight away read in. So even if you wanted to even simplify this further, you could literally just replace this with that read table command, and it'll just in one line bring it all into a scRNA object. But you don't have to do that. Okay, I think that is enough for one day. I don't see any questions in the chat, so hopefully if you've got any questions, you'll email me later, or stop by office hours if you run into any issues. But if not, then thank you all for sticking with me for such a long session. I know that we went way over time, but I'm glad that we were able to cover everything that I had planned for us to cover today, because so much of this is so fundamental to the rest of what we do with single cell. Okay, next time is going to be shorter. Thank you for sticking with us, and thank you all for attending. Next time, we're going to pick it up with PCA and nearest neighbor graphs, along with clustering and low-dimensional embedding. So this is going to be the meat of the analysis, and we're going to do that next time. So with that, thank you all for being here, and I will see you all next week. Have a good one.